Unit 2 is a very dead chapter to revise, but it's fairly basic stuff. Cell theory has 3 parts. Both plant and animal tissue are made of cells. Cells are the basic unit of life, and cells can only come from existing cells. Okay, you know what? Forget all of that. All you need to know for 2.1 to 2.3 is the following. Okay, first up, learn about the different mounts. Now, there's a whole bunch of different mounts, but really, all you need to know is what a wet mount is, what a smear slide is, and what sectioning is. A wet mount is basically when you put a droplet on the slide, put the specimen in the droplet, then place a cover slip on at 45-degree angle to prevent air bubbles from being trapped. Sectioning is basically cutting specimens into small slices using a sharp blade called a microtome. Also, learn the mark scheme for why you stain things. If it comes up in an exam, it will be a 2 or 3 mark question. See, disadvantages of staining would be you kill the thing. That's pretty much it for 2.1. Now for 2.2, it's literally just basic maths. If you're terrible at maths, as in you got less than, I don't know, a 7 at GCSE, do a couple exam questions, you should be fine. All it is basic ratios and rearranging a formula. And learn the definitions of magnification and resolution. That's it for 2.2. For 2.3, all you're going to need to do is memorize this table comparing all the different microscopes, and know a couple things about electron microscopy.
That's amazing. This is the most advanced electron microscope on the eastern seaboard. It's unreal.
So it works by electromagnets, focusing a beam of electrons to illuminate the specimen. The reason why it shows more detail than light microscopes, because electrons have a much smaller wavelength than light waves.
Ooh, that's kind of small. Yikes.
Learn the disadvantages of electron microscopy. It's expensive, it needs a carefully controlled environment, specimens can be damaged by the electron beam, and specimens need to be dead. You need to learn what artifacts are. Artifacts are structures that are produced due to the preparation process. Also memorize this table comparing light and electron microscopy. It might be a 6 marker. And that is pretty much it for 2.1 to 2.3.